everybody. Welcome back to Succinct Psychology, or Psychcinct. My name is Prescott, and we're going to continue Unit 3 Biopsychology from the OpenStax online textbook, Psychology 2E. And let's go ahead and keep it on. So last time, went over gene-environment interactions, Punnett squares, phenotypes, genotypes, theory of evolution. And so today, we're going to continue on and we're going to talk a little bit about neuron structure and axon cells, all that fun stuff. So, continuing on from last time, we have neuron structure. So the function of a neuron is aided by specific aspects of its structure. Its semi-permeable membrane allows smaller molecules or molecules without an electrical charge to pass through it while stopping larger or highly charged molecules. Incoming electrical signals from other neurons are received by the dendrites, which then send the signal down the axon. So here we have a figure. This illustration shows a prototypical neuron, which is being myelinated. So axons are covered in a myelin sheath made of a fatty substance that insulates axons and allows the signal to travel down the axon quicker. At the end of the axons are terminal buttons, which contain synaptic vessels, so storage sites for chemical messengers called neurotransmitters. So here we have the axon, you can see the myelin sheaths, and then these terminal buttons, which contain the synaptic vessels, and we will go into what the synapse is right here. So the synapse is the space between the terminal button of one neuron and the dendrite of another. And so you can see here the terminal buttons, these are the dendrites of another neuron, so the synapse is that space in between the two. In this pseudo-colored image from a scanning electron microscope, a terminal button, green, has been opened to reveal the synaptic vesicles, orange and blue, right here, inside. Each vesicle contains about 10,000 neurotransmitter molecules. So, pretty interesting. Now, at resting potential, Na positive, these blue pentagons, are more highly concentrated outside the cell in the extracellular fluid, shown in blue. Whereas K positive, purple squares, shown here, are more highly concentrated near the membrane in the cytoplasm or intracellular fluid. So extra is outside the cell, intra is in. Other molecules, such as chloride ions, these yellow circles, seen here, and negatively charged proteins, big brown squares, help contribute to a positive net charge in the extracellular fluid and a negative net charge in the intracellular fluid. So it's positive, negative. So going on, we have action potential. So neurotransmitters from nearby neurons attach to receptors on dendrites, causing the membrane potential to change. So depolarization is when the membrane potential becomes less negative, making the neuron more likely to fire, which is called excitation, making it more likely to fire, exciting it. Hyperpolarization is when the membrane potential becomes more negative, making the neuron less likely to fire, which is inhibition. It's making it less likely to fire. Excite, more likely. Inhib, less likely. If the level of charge reaches the threshold of excitation, an action potential will occur. Ion channels open, causing Na positive to rush into the cell and the inside to momentarily become more positive. So threshold of excitation is a level of charge in the membrane that causes the neuron to become active. And action potential is an electrical signal. So action potentials act on an all or none principle. The incoming signal is either sufficient to reach the threshold of excitation or it is not. So there's no halfway reaching the threshold. It's all or nothing. Here we have a little graph. During the action potential, the electrical charge across the membrane changes dramatically. As you can see here, you start with your resting potential, reach the threshold of excitation, you reach your peak action potential, and then you start to repolarize and go into hyperpolarization, which is again making it less likely to fire. Now we have reuptake. So once an action potential has occurred, excess neurotransmitters in the synapse either drift away, are broken down, or are reabsorbed. So reuptake involves moving a neurotransmitter from the synapse back into the axon terminal from which it was released. So reuptake involves moving a neurotransmitter, from the synapse back into the axon. Oh, it just repeats itself, that's weird. So here you can see the process of reuptake, neurotransmitter goes back into the synapse so it can be released again between the terminal buttons and dendrites. So what are neurotransmitters? So neurotransmitters is a chemical messenger of the nervous system. Different neurons release different types of neurotransmitters that have many different functions. So from a biological perspective, it's a view that psychological disorders like depression and schizophrenia are associated with imbalances in one or more neurotransmitter systems. So here are some neurotransmitters. We have acetylcholine, which dictates muscle action and memory, beta-endorphin, which is pain and pleasure, dopamine, which is mood, sleep, and learning, norepinephrine, heart, intestines, and alertness, and finally serotonin, which is mood and sleep. So there are some drugs for neurotransmitters. So first we have psychotropic medication, so drugs that treat psychiatric symptoms by restoring neurotransmitter balance. So an agonist is a drug that mimics or strengthens the effect of a neurotransmitter, while an antagonist is a drug that blocks or impedes the normal activity of a given neurotransmitter. So easy way to remember that: antagonist of a story is trying to block or impede the hero, so do antagonist drugs. But they're doing it with neurotransmitters, not heroes of a story. So agonist/antagonist drugs are prescribed to correct neurotransmitter imbalances. Example, Parkinson's disease is associated with low levels of dopamine. Dopamine agonists are often prescribed as one form of treatment. So again, dopamine is mood, sleep, and learning, so it's trying to strengthen the effect of the dopamine. While schizophrenia, on the other hand, is associated with too much dopamine. So many antipsychotic drugs are therefore dopamine antagonists. They're trying to impede the normal activity of that dopamine neurotransmitter because there's too much of it. So going on, we have the parts of the nervous system. So the nervous system is divided into two major parts: the central nervous system, shown here is the brain and spinal cord, and the peripheral nervous system, which is all the different nerves. So the peripheral nervous system is made of two different parts: the somatic nervous system, which relays sensory and motor information to and from the central nervous system, and the autonomic nervous system, which controls our internal organs and glands and can be divided into the sympathetic and parasympathetic nervous systems. So the sympathetic nervous system is involved in stress-related activities and functions, and it prepares us for fight or flight. Now, I'm sure you've heard of the fight or flight response, but just to give you a definition, is an activation of the sympathetic division of the autonomic nervous system, allowing access to energy reserves and heightened sensory capacity so that we may fight off a given threat or run away to safety. So, the sympathetic nervous system, it triggers your fight or flight response in case, you're in a dangerous situation and you either need to be able to defend yourself or get out of there. Now, the parasympathetic nervous system is associated with routine, day-to-day operations of the body under relaxed conditions, and also has the rest and restore response, which relaxes the body after fight or flight, aka rest and digest. So sympathetic nervous system gets you ready for fight or flight, and parasympathetic calms you down after you're out of that situation. So the sympathetic and parasympathetic nervous systems complement each other to maintain homeostasis, which is a state of equilibrium in the body. Here we have subdivisions of the nervous system. So starting off, you have the nervous system, which can be divided into the central nervous system, brain and spinal cord, and the peripheral, which is nerves to and from your central nervous system. Now, the peripheral can be broken down more into the somatic and autonomic nervous systems. So somatic is touch, hearing, etc., and muscles that move the body. And then autonomic are the internal senses for gut, smooth muscles and gut, etc. And autonomic has two more branches, which is the sympathetic, emergency, and parasympathetic, which is housekeeping. So just relaxed state, day-to-day stuff. So we're going to stop this video here. We will continue to go into the nervous system, brain and spinal cord, all that fun stuff in the next video. Again, my name is Prescott here at Psychcinct, Succinct Psychology, and I hope that you guys tune in for the next video. Have a great day, and see you next time.